The equipment needed in this experiment is a voltmeter, 2 beakers, wires, a strip of copper sheet, a strip of zinc sheet, sandpaper, copper sulphate and zinc sulphate solution, filter paper, thin metal tongs and a salt solution. A voltaic cell is an electrochemical cell that uses a chemical reaction to produce electrical energy. The important parts of a voltaic cell are the anode, the cathode, and the salt bridge. The anode is a positive electrode on the left and is an electrode where oxidation occurs, whereas the cathode is a negative electrode on the right is an electrode where reduction occurs. With a strip of metal, it is important to clean the surface of them with some sandpaper to ensure that they are both shiny, as there may be a dull layer on top. It may take some time, however, it is definitely important to ensure that the metals can work properly. Once you have cleaned both the strip of copper and zinc, you then want to get your beakers and wires. Make sure to add crocodile clips to one end of both wires. Carefully place the metal strips inside the beakers by clipping them to the beakers with the clips on the wires. These metal strips are the electrodes of the voltaic cell that we will create. As the metal strips are now in place, you want to make sure to get your beaker with the copper strip, which has a more bronze colour, and get your container with the copper sulphate solution, which has a nice blue colour. Carefully pour some of the copper sulphate solution into the beaker so that most of the strip is submerged, but make sure that the crocodile clip is not in contact with the solution. Make sure again not to have the crocodile clip in contact with the solution, but to have most of the strip submerged. As there is a gap between both electrodes and solutions, the circuit is not complete. To complete this circuit, we need to add a salt bridge. With your salt solution and filter paper, you use your thin metal tongs to carefully dip the filter paper into the solution, and then put either end of the filter paper in both of the beakers with the electrodes. Now the circuit is complete. The final step is to actually measure the cell potential, which is measured in volts. We get our voltmeter and set it to make sure it has enough decimal places. Make sure to watch carefully to see where we have plugged the wires into. We then use the wires clipped onto the electrodes and plug them into the voltmeter. As you can see, the voltmeter has a reading of around 1.044 volts. Now to dispose of the solutions. Please ask your lab technicians how they would like the solutions to be disposed of, but we are going to be using a waste beaker for each of the different solutions. Please do not pour the solutions of heavy metal ions down the sink, but instead place all residues in the waste beakers in a fume cupboard.